Protein 3D structural database PDB. What is PDB? PDB is a database containing experimentally determined three-dimensional structure of protein, nucleic acid and other biological macromolecule. NMR, X-ray crystallography and electron microscopy are the three techniques to determine the three-dimensional structure of this biological macromolecule experimentally. After determining the structural data, the data is submitted to PDB. Okay. So what is PDB? A PDB is a database for the three-dimensional structural data of large biological molecules such as protein and nucleic acid. The data typically obtained by X-ray crystallography, NMR spectroscopy, and increasingly by cryo-electron microscopy. The data is freely accessible on internet by the website of its member organization, that is PDBE, PDBJ, RCSB, EMDB and BMRB. These all are the member organization of PDB. The PDB is overseen by the organization called WPDP, that is World Wide Protein Data Bank. The mission of WPDP is to maintain a single protein data bank key of micromolecular structural data that is freely and publicly available to global community. Why did it start? Why the PDB has been started? The two forces that converge to initiate PDB are the growing crystallographic data determined by X-ray diffraction and the development of BRAD in 1968. BRAD is a molecular graphic display used to visualize the three-dimensional structure of protein. So these two are the forces that converge to initiate PDB. First is the growing collection of protein structure determined by X-ray diffraction, and second is the development of BRAD, the molecular graphic display used to visualize the protein structure in 3D. Coming to the history, in 1969, Dr. Mayer began to write the software to store the atomic coordinate files in a common format to make them available for geometric and graphical evaluation. In 1971, Dr. Mayer's program called SEARCH enabled networking that enabled the researchers to access information from the database to study the protein structure offline. In 1973, upon the Hamilton's death, Dr. Tom took over the direction of PDB for 20 years. In 1980, IUCR guidelines established number of structure deposited increases and independent biological database such as NDB was established. NDB is a nucleic acid data bank. In 1998, PDB was transferred to Research Collaboratory for Structural Bioinformatics, that is RCSB, and complete transfer since 1999. Dr. Berman of Rutgers University was the new director. In 2003, with the formation of WPDB, the PDB became an international organization having the three-member organization, that is PDBE. PDBE means PDB Europe, PDB Japan and RCSB. These three are the member organization of WPDB in 2003. In 2006, BMRB joined PDB, and in 2012, EMDB joined PDB-RB. So presently, PDB has a 5-member organization. They are the PDBE, PDBJ, RCSB, BMRB and EMDB. Who runs WPDB? The member organization of WPDB are the PDB in Europe, PDB Japan, Research Collaboratory for Structural Bioinformatics Protein Data Bank, RCSB, Biological Magnetic Resonance Data Bank, BMRB, and Electron Microscopic Data Bank, EMDB. These all are the member organization of WPDB. Since 1971, the PDB has served as a single repository of information about the three-dimensional structure of protein, nucleic acid, and complex assemblies. The WPDB organization manages the PDB key and ensures that the PDB is freely and publicly available to global community. This is the home page of WPDB. See, it contains the validation tool by which you can validate your 3D structure of the protein determined by NMR or by electron microscopy. To deposit your structure, you can click on the deposition. You can see the directories where there is a micromolecular directory, small molecular directory, peptide like antibiotic and inhibitor molecular directories. You can see the documentation. If you want to study about WPDB, you can go to About Us. Coming to PDBE, that is Protein Data Bank in Europe, contain a rich information about all the PDB entries, multiple search and browse facilities, advanced services including PDBE PISA. PDBE PISA is a tool used to predict the quaternary structure of the protein. Then it also contains the PDBE fold. PDBE fold is the structural alignment tool in which you can go for both the pairwise and the multiple alignment of protein structure. Similar to that of the BLAST and Clustal W. In BLAST, what we go for the pairwise alignment of the sequence to find the part which is common in both. In the Clustal W, we go for the multiple sequence alignment tool to find the conserved part in the sequence. Similarly, in the PDBE fold में हम क्या करते हैं? We compare the structure. We align the three-dimensional structure of the protein to find the conserved part in the protein structure. Okay, so PDBE fold is the structural alignment tool where we can perform both the pairwise and the multiple comparison of protein structure. Then advanced visualization tool and the validation tool of NMR and electron microscopic structure. It contains the visualization tool by which we can visualize the 3D structure of the protein. We can validate the structure determined by NMR and the electron microscopy. There are various validation tools are also in PDBE, then tools for bioinformaticians. See, this is the homepage of PDBE, Protein Data Bank in Europe. These all are the services provided by PDBE. Next is the PDBJ, that is the PDB in Japan. Supports browsing in multiple languages such as Japanese, Chinese, and Koreans. Seesaw identify the functionally and evolutionary conserved motif by locating and annotating the sequence and structural similarity. Seesaw is similar to that of PDBE fold. It is also a structural alignment tool in which we can go for both the pairwise and the multiple alignment of protein structure. Now, to find the conserved motif by locating and annotating the structural similarity. The full form of Seesaw is sequence derived structural alignment weight. Okay, it is also the structural alignment tool. And it contains the tools for bioinformaticians and more. See, this is the homepage of PDBJ. From here, you can select the language, and this all are the services provided by PDBJ. Coming to RCSB PDB, a simple and advanced searching for micromolecules and ligand, tabular reports, specialized visualization tool, sequence structural comparison tool, RCSB PDB mobile, molecule of the month, and other educational resources at PDB 101 and even more. So this is the homepage of RCSB PDB. Here is the search engine. This is the molecule of the month. To deposit your structure, you can click here. To retrieve the information from RCSB PDB, you can click on the search. You can type here. There are various visualization tool like JMOL, RESMOL, by which you can read the PDB file. You can create the custom picture of your protein. Like you can change the bond angle, color of the bond. You can change the format, like you can make the structure in ball and stick model or in the ribbon model. Okay, तो visualization tools से हम क्या करते हैं? We can read the PDB file. ठीक है, बहुत सारे visualization tools होते हैं जैसे कि आपका JMOL, RESMOL वगैरह है, which help to read the PDB file, and you can create the custom picture of your protein. Then there are various analyzing tools which help to measure the bond angle, the distance, है ना, of your protein structure and various structural feature also. You can download the structure of your interest and you can learn more. See the PDB 101. The PDB 101 is the online portal for the teachers, students to explore the protein structure. This is all about RCSB PDB. Then BMRB, the BMRB collects the NMR data from any experiment and capture assigned chemical shift, coupling constant and peak list for the variety of micromolecules, contain derived annotations such as hydrogen exchange rate, pKa value and relaxation parameters. Okay, BMRB जो है वो कहाँ से collect करता है data को? Collects data from NMR data, okay, from any experiment and capture chemical shift, coupling constant and peak list of the variety of micromolecules. See, this is the home page of BMRB and this all are the services provided by BMRB. Then EMDB collects the three-dimensional volume and associated information of micromolecular complexes and subcellular structure from electron microscopy and electron tomography. This thing you have to remember. In the EMDB, collects data obtained from electron microscopy and electron tomography, and in BMRB collects data from NMR. Okay, develops resources for searching, data mining, analyzing, validating, and visualizing data. Okay. This is the home page of EMDB. Here is the search box. How the data is collected. The PDB is a repository of atomic coordinates and other information describing the protein and other important biological micromolecules. The structural biologist uses methods such as X-ray crystallography, NMR spectroscopy, and cryo-electron microscopy to determine the location of each atom relative to each other in a molecule. They then deposit this information, which is then annotated and publicly released into the archive by WPDP. Okay, so what the structural biologist will do? You know, the protein, nucleic acid, carbohydrate, the biological macromolecules, they does not have any real image. You know, if you see the protein under the microscope, you didn't find any structure like a cell. जैसे अगर अपन बैक्टीरिया को अगर माइक्रोस्कोप के अंदर रखते तो हमको एक स्ट्रक्चर दिखाई देता है, ठीक है। जो प्रोटीन है, कार्बोहाइड्रेट है, उनका कोई स्ट्रक्चर नहीं होता, उनका कोई रियल इमेज नहीं होती है, ठीक है। तो व्हाट द स्ट्रक्चरल बायोलॉजिस्ट विल डू, दे determine the location of each atom respective relative to another in the molecule. Structural biologist क्या करते हैं, atoms जो मॉलिक्यूल है, प्रोटीन इज अ मॉलिक्यूल, ठीक है, तो उनमें एटम के बीच के रिलेशन को निकालते हैं। एक एटम दूसरे एटम से कौन से बॉन्ड से जुड़ा हुआ है, उनके बीच में बॉन्ड लेंथ क्या है, बॉन्ड एंगल क्या है, अट्रैक्शन रिपल्शन क्या है, ठीक है, वो ये सारी इंफॉर्मेशन कलेक्ट करते हैं बाय यूजिंग एक्सरे क्रिस्टलोग्राफी, एनएमआर और बाय इलेक्ट्रॉन माइक्रोस्कोपी. After collecting all this data, they model the structure of the protein. They model the structure of the protein in such a way which reflect the experimental data in a consistent way by providing information about the partial arrangement of a group of atom in a molecule. और ये जब सारे डेटास कलेक्ट करने के बाद वो प्रोटीन के स्ट्रक्चर को मॉडल करते हैं. अगर दो एटम के बीच में ये डिस्टेंस है, ये बॉन्ड एंगल है, तो उसका स्ट्रक्चर ऐसा हो सकता है, ठीक है. ऐसा स्ट्रक्चर वो डालते हैं कि हमको उस स्ट्रक्चर से वो एटम्स का अरेंजमेंट समझ में आए. फॉर एग्जांपल, इफ आई से दैट हियर देयर इज अल्फा हेलिक्स, यहाँ पे अल्फा हेलिक्स है, तो हमारे माइंड में अपने आप ही पिक्चर बन जाएगा कि अल्फा हेलिक्स मतलब कि हाइड्रोजन बॉन्डिंग है, और वो हाइड्रोजन बॉन्डिंग जो है वो मेन चेन में है ना, NH और CO ग्रुप में, तो वहां पर अल्फा हेलिक्स आएगा, एक स्पाइरल लाइक शेप आएगा, ठीक है. अगर मैं बीटा स्ट्रैंड बोलती हूँ, अगर कहीं पे बीटा स्ट्रैंड है, तो यू आपके माइंड में अपने आप ही पिक्चर आ जाएगा. अगर बीटा स्ट्रैंड है मींस हाइड्रोजन बॉन्डिंग जो है वो आपके नेबरिंग पॉलीपेप्टाइड सेगमेंट्स के बीच में, मेन सेगमेंट में नहीं, नेबरिंग में है. पैरेलल है मतलब कि CO और NH जो है वो सेम साइड में है, अगर अपोजिट है तो वो एंटी पैरेलल कहलाएगा। ठीक है, ये हो गया सेकेंडरी स्ट्रक्चर। अगर मैं टर्शरी स्ट्रक्चर बोलती हूँ तो वहां पर हम डाइसल्फाइड बॉन्ड, सॉल्ट ब्रिज, है ना, वो सारी अरेंजमेंट भी आ जाती है। तो जो अल्फा हेलिक्स है, बीटा प्लेटेड शीट है, ये सब जो है वो मॉडलिंग की गई है, स्ट्रक्चर की मॉडलिंग की गई है। एक्चुअली में जब आप प्रोटीन को देखते हैं तो जस्ट आपका वहां पे मॉलिक्यूलर एटम्स का अरेंजमेंट है, वो वैसा दिखता नहीं है। ठीक है, इट इज जस्ट अ मॉडल विच रिफ्लेक्ट द एक्सपेरिमेंटल डेटा। ओके, so after creating all this information, they deposit the information to the WWPDB. The content of WWPDB, the PDB database is updated weekly along with its holding list. As of 1st April 2020, the PDB comprised of this many structure. How to retrieve data from PDB? RCSB PDB website allow you to search and explore the information under PDB header, including information on experimental method and chemistry and biology of the protein. To search any data in PDB, we have to go to RCSB PDB. There is the search box where you can from where you can search. Once you found the PDB entry that you are interested in, you may use the visualization program to allow you to read the PDB file, display the protein structure on your computer, download the information, and create the custom picture of it. Once you get your PDB entry, you can use the visualization program. Visualization program जैसे कि मैंने बताया था, जेमॉल, रेसमॉल, बहुत सारे visualization program होते हैं which help you to read the PDB file, display the protein structure on your protein on your computer, and you can create the custom picture of it. You can change the color of the bond, you can change the format of the protein structure, like you can make the ball stick model or you can create the ribbon model. अलग अलग टाइप से आप वहाँ पे बना सकते हो. This program also often include the analysis tool that allow you to measure distance and bond angle and identify the interesting structural feature. There are various analysis tool also by which you can measure the distance and the bond angle and also identify the interesting structural feature. And how to search? One can search for a protein of interest by using the search bar in RCSB PDB website. Search box में अपन search करते हैं. Here you can it allow one to search either by typing the PDB ID or by the author name or by the sequence of the protein or any particular ligand of interest. If you know the PDB ID, you can type here. If you know the author's name, you can search by the author name or you can search by the sequence. Okay. What is PDB ID? Okay, the PDB ID is similar to that of the accession number, which we have studied in the sequence database. What is accession number? They are the unique identification number given to the sequence record in the database. है ना, न्यूक्लिक एसिड सीक्वेंस डेटाबेस हमने पढ़ा, प्रोटीन डेटाबेस हमने पढ़ा, तो हर सीक्वेंस को एक आईडी दी जाती है जिसको हम बोलते हैं एक्सेशन नंबर. Similarly, in the PDB database, the PDB will provide the PDB ID. Okay, the PDB ID is a 4-character unique identifier of every entry in protein data bank. The 4-character PDB ID is assigned to each new structure at the time of deposition. The first character is numerical in the range of 1 to 9, while the last 3 characters can be either numerical in the range of 0 to 9 or a letter in a range of A to Z. If the PDB ID of any entry in a PDB data bank is known, it is the most direct way to retrieve it from the database. If you know the PDB ID of the protein you are interested in, it is the most direct way to retrieve it from the database. One or more PDB ID can be typed, copied, or pasted in the search box. You can search either by single ID or by the multiple ID. The multiple IDs are separated by comma or space, including the break line. Okay? जैसे यहाँ पे मैंने एग्जांपल में दिया है, if you want to search for 4HHB, you can type it on the search box and click on the search. It will open up this page. See, this is the biological structure of 4HHB. This is the paper in which the structure has been published. Then this is the DOI. classification, organism name, mutation, the date at which the sequence was deposited and released, the deposition author's name, and here is the experimental data, the method by which the structure has been determined, resolution and R value. You can also search by the multiple IDs. If you are searching by multiple ID, you have to uh, use comma or space between the two ID. And after clicking on search, it will open up this page. See, then whatever structure you want to study in detail, either you can click on the image or you can click on the ID. Then what to learn future, PDB 101 is the online portal for the teacher, student and general public to promote exploration in the world of protein and nucleic acid, learning about the diverse shape and function of this biological micromolecule, help to understand all the aspects of biomedicine and agriculture, from protein synthesis to health and disease to biological energy. So this is all about PDB.